So, hello students, welcome to the lecture 5. So today we will be discussing with the polymerase chain reaction. In my previous lecture, one of the previous lectures, I have given a little bit of description about the PCR. So this is a polymerase chain reaction, which is a biochemical process that's amplifying a single DNA molecule into millions of copies in a very short time. So the PCR is actually involved in all of the modern processes of the molecular biology. Whatever the experiment process is coming, PCR is actually involved there. And this reaction is very simple to set up and also very cheap. So as well as that, this is a very robust, speedy, and flexible and sensitive experiment to do. So the only requirement for doing a PCR is some kind of knowledge about the DNA sequence of the target fragment required to initiate a PCR experiment. So the PCR actually uses a temperature-dependent cycling process. So where the temperature is actually used to proceed to the experiments and has a various steps that has been catalyzed by the enzyme and enzyme that is the DNA polymerase, which actually doing the DNA synthesis. So every cycle in the PCR, they consist with the 3 steps. Firstly, the denaturation. So the denaturation of DNA template is happened by heat, usually is more than 90 °C. And then in the next step, we do have an annealing where the primers, which is a small oligonucleotide, they normally uh, having a length of between 20 to 30 base pairs, but it's vary depending on the experiment. So the primer, they bind to the annealed, that is bind to the single-stranded DNA, that is the denatured double-stranded DNA to make a single-stranded DNA, and the primer is bind to one of those single-stranded DNA. And then in the third step, that happens the extension where the DNA polymerase actually synthesize the double-stranded DNA. And this process is actually repeated for 25 to 35 times, depending on the target size of the product. So if we have a long DNA, normally it's doing around 30 cycles, but you can do a 35 cycles if you have a really long PCR product. So that's how the PCR experiment is doing. So the first step is the denaturation, where the double-stranded DNA has been denatured to make a single-stranded DNA. In the second step, the annealing of the primer then annealed to the single-stranded DNA. And in the third step, this is extension or synthesis of the DNA molecule, double-stranded DNA from that single-stranded DNA by DNA polymerase. And in that way, we have actually getting a millions of copies of same DNA fragment that we start at the beginning. So PCR cycle conditions is, as I said, that the initial denaturation is normally done in more than 90 degrees centigrade. So normally using a 95 degrees centigrade, then it depends on maybe 2 to 5 minutes. And this is just a one cycle. And then start the denaturation. It can be 94, 95 degrees centigrade, and it's actually going for 15 to 60 seconds. So this is just a general representation of a PCR cycle. It will vary depending on the amplicon size, that is the final product of the PCR. And then the annealing is also dependent on the primer, because the primer is actually binding to the single-stranded DNA. So it also, the annealing also depends on the melting temperature of a primer. So that we will be coming into later slides. So the annealing normally done in around 50 to 65, 66 degrees centigrade, depending on the melting temperature of the primer. And it can be done in 30 to 60 seconds. And in the next step is the extension. This is normally done in 72 degrees centigrade. And it is depending on, again, the size of the final product. So if you are doing a PCR of 500 base pairs, you might do an extension for only 15 seconds or 30 seconds. If you do a 4 kb base pairs of a amplicon or final product, you might do a 4 minutes. Is a general rule is a 30 seconds per kb or a 1 minute per kb. So that's how the PCR is actually set up. And there is a final extension step as well, where all the 25 to 35 repeated cycle has been finished. Then you just give the time to for the final. Is there any other extension need to be done? You maybe give it like 5 minutes to 10 minutes, again, depending on the size of the fragment. And then you can just hold the whole the experiment into 4 to 12 degrees centigrade for infinite times, or you just take out the whole experiment as soon as it's finished and run a gel. So it all depends on how you are setting up the PCR. So this is just a general representation of a PCR experiment. So the next is what was the difference between the PCR and the DNA replication? So the PCR is you need a heat to actually melt the DNA or to denature the DNA. That is making a double-stranded DNA to a single-stranded DNA. But in case of DNA replication, the cell machinery use the helicase and the single-stranded DNA binding protein and topoisomerase to denature the double-stranded DNA. Polymerizing the DNA, you use normally the DNA Taq polymerase, which is commonly used in PCR product, whereas in the cell it's normally the DNA polymerases. Primers, the primer we actually commercially made the primer, which also called the oligonucleotide, and this has been added to the reaction mixture. On the other hand, the primers is used in the DNA replication by the cell machinery. And the NICs, it is not applicable because this is the fragments. So normally they have a blunt end in the PCR products that normally don't produce the overhang as we have seen in the restriction digestions. So PCR product always produce a blunt end fragment. On the other hand, DNA replication, the ligase is used, which we have seen in the restriction digestions, where the vector and the DNA are producing a overhang, single-stranded overhang that has been joined together by the ligase. So this is again a typical PCR mixture. So what are the components you need to do a PCR experiment? It's normally the DNA template, and it's very depending on which organism you are using to do a PCR. If you use a plasmid DNA for a PCR, it might be 1 picogram is more than enough to do an experiment, PCR experiments, provided that the plasmid have a good concentration. The extracted plasmid have a good concentration. For like a fungi or the yeast, you can use like a 1 nanogram to 5 nanograms. For the mammalian DNA, it might be a little bit more, but it's always a variable. Then we need to use the DNTP. So DNTP is that 4 bases. That's the building blocks. We need those DNTPs, and they are just using a final concentration about a 200 micromolar. We need a PCR buffer and a magnesium chloride. We will discuss it a little bit later. And definitely the primers. We need a 2 primer. One is the forward primer and the reverse primer. So that one binding to the forward forward strand, and another one is the reverse strand. Then we also needed DNA polymerase and a water. I should say it's a molecule, which is a really a deionized, very purified water. So PCR experiment or any other experiment, sometimes it's very important about using the clean water. And normally in the lab, we use a water which is called a molecule, which is a deionized water, and it's a really a pure form. So it makes a final volume of 50 microliter, but this is just a representative. You can scale down your PCR experiment to 10 microliter or 20 microliter or 30 microliters, depending on how many microliters of final volume you really need to run a gel. So I normally do a 10 microliter reaction in just to check the plasmid or confirming a DNA insert into the plasmid, and then use it that 10 microliter after the PCR. I got a maybe 10 or 9 microliters, and then run that 9 or 10 microliters into the gel to check that my DNA fragment is present or not. But in some cases, you might need a more volume, then you can use a 50 microliters, or even you can use 100 microliters. So you can actually manipulate those scaling up or scaling down depending on your experiment. So just check what are the components we are using. So what is the actually of their roles into the PCR experiment. So the DNA template, this is actually contain the target sequence. And normally the double-stranded DNA has been used for the PCR reaction, but single-stranded can also be used. And the amount of, as I say, the amount of template is always varies from picogram to nanogram level, such as the plasmid, you can use just a 1 or 10 micro picogram of plasmid DNA is sufficient to check either the plasmid or the DNA fragment. Then we can use we need to use the buffer with the magnesium chloride. So buffer is actually used for the pH of the reaction. And magnesium chloride has a very important role into the PCR experiment. So all of the thermostable DNA polymerase, they require a free divalent cation. And that is usually the magnesium for its activity. So this magnesium actually reacting with the DNTP to form a complexes that are substrate for the DNA polymerases, and also stabilize the primer and the template complex. So magnesium definitely has a very important role into the PCR reaction, and they normally actually come with the buffer. So the buffer and the magnesium chloride is normally coming as the same tube. Then DNTP, which is the building blocks, as I said, that this is the nucleosides like the adenine, guanine, cytosine, and thymine, and that is used for the synthesis of the DNA. Primers. So primer is another critical component of the PCR. So designing of that a primer is very important. Efficient primer is very important to get the efficiency or the specificity of amplification reaction. So that is where I said that for the PCR, there is a one thing we need to know about some of the DNA sequence of the target fragment. So we need to know to design the primer so that the primer can bind the single-stranded DNA, and from there DNA polymerase can. And this primer, there are a number of factors need to be considered when designing a primer, such as the guanine and cytosine content. So it is ideally said that the guanine and cytosine content can be between 40 to 60%. So if any of the primer having a more guanine and cytosine, so they can form a secondary structure, and they don't then bind to the single-stranded DNA, which we really want. Then the primer should not be self producing like a self-complementary. So if the primer, both the primers are self-complementary, then they will bind together and form a primer dimer. In the melting temperature, as I said, that is important for annealing. So it is actually used for the annealing temperature of the PCR reaction. So normally when there is a formula for melting temperature, you can go and have a look. Normally the melting temperature is used to set up for the annealing temperature of a PCR reaction. and it is between the 55 to 60 is giving a best result. But depending on, so if you have a long primer, which may be 40 base pairs, your melting temperature will be high. If you have a like 25 to 30 base pairs of melting primer, then your melting temperature will be a bit lower, which is maybe in between the standard zone. And polymerase, definitely, we need a PCR polymerase, which is really a thermostable polymerase, and the Taq-DNA polymerase is commonly used in the PCR reaction, but there are other polymerases also being used, like depending on the how proof or how fine the PCR product you are looking for, and the ability to synthesize a large DNA fragment. And obviously, the final step is the sterile water, which is normally the molecule, as I said, the deionized water, and which has been sterilized before using that part. So if you have all of the component is right and good amount, and but if your water is contaminated, then your PCR won't be working. So water is a very, very important component of the PCR reaction.